Good evening, everyone. The boat is in. It's Wednesday, and we're at Ocean Club. Welcome to See and Learn. Tonight, we're at a special venue. Saba's new restaurant is Ocean Club. And tonight's speaker is Dr. Erinn Muller. Sounds a little doom and gloom, but she's going to give us hope about coral disease, and specifically Skittle D. So, thank you for watching abroad or live on Saba, and we'll be back before long to bring you tonight's venue at Ocean Club.
And one of the ways that you can support us is to leave us a review on great nonprofits. Lynn and I also have these business cards now, so we're official, that have the Great Nonprofits logo on. So if you want to spend 3 to 5 minutes and just say how much you enjoyed our program, it really helps us out with our funding. So that would be great. And then upcoming events. Tomorrow we have 2 field projects starting at 1:30. We have a dive with Dr. Muller. There are a few spots still open. You can sign up in the back with Alexis. And then there is also a night hike tomorrow night with Dr. Lauren Esposito, who is in the back. Give a wave. We'll be looking for scorpions, spiders, and all kinds of fun stuff. So you can sign up, and if you do any of our field projects, you win a free hat. So definitely do it. And then Friday is our next presentation at Lollipops with our land crab expert, Bill Bigelow, who is also in the audience here. Yes. So you can join him 5:30 at Lollipops. And then tonight's speaker, how many people are divers or snorkelers here? Yes? How many of you snorkeled or went diving 10 or more years ago? Okay, good majority, yeah. So you can compare the coral reefs of the past to the present then. Most of you will probably agree that our reefs are in a little bit of trouble. But tonight is not all about doom and gloom, but instead on what can be done. Dr. Erinn Muller is a senior scientist at Mote Marine Laboratory and manages both the coral health and disease and coral restoration programs. Dr. Muller's research focuses on identifying corals that are resilient to major threats, such as climate change and coral disease. For the last several years, much of her research has been associated with the newer stony coral tissue loss disease, the most devastating disease that coral has ever been documented. Erinn has studied coral health and disease for over 18 years, and in that time has published more than 50 papers and mentored more than 50 students, and has been the recipient of prestigious awards. To make sure you have a keen eye on your next reef experience, let's learn a skittle bit about a not-so-sweet subject with Dr. Erinn Muller.
Thank you so much for joining us tonight. It's an incredible pleasure to be able to share with you a little bit about the research that we've been doing on the different types of treatments that we can use to fight coral diseases. So this is definitely a story about corals and corals lost, but it's also a story about the novel collaborations that occur when we're faced with crises. And that's kind of where we are today with the state of coral reefs. But there are many different innovations that come out of really stressful and hard situations. And I'm here to share with you just one of those situations that I've been able to integrate my research into over the last several years. So I just want to take the time to recognize my collaborators on this slide. Dr. Sarah Williams, she is my postdoc, and she definitely is the foundation of a lot of the work that I'm going to be sharing with you today. And my collaborators, Katie Curtis and Mike Favero from Ocean Alchemists, which is a unique startup company in Tampa, Florida, that has allowed for a lot of these treatments to start to be implemented within the reef environment. So Dr. Williams and I come from Sarasota, Florida. We are staff members at Mote Marine Laboratory, which is one of the last remaining completely independent nonprofit research institutes within the United States that focuses solely on marine conservation and research. We have over 200 different staff, 30 different PhDs focusing on 24 unique programs facing our oceans today. Now, the program that we primarily work with is the Coral Health and Disease Program and also the Coral Restoration Program. And much of that work takes place here in Summerlin Key, Florida. This is our sister campus, the International Center for Coral Reef Research and Restoration on Summerlin Key. It's an incredible building that looks kind of prison-like, but within those walls is a really collaborative sense of innovation. We have classrooms there, lab space, and we actually are surrounded by the U.S.'s largest coral restoration nursery within this infrastructure too. And this team works together to study corals and coral reefs because we think it's an incredibly important ecosystem and one that's facing critical threats. So why do we spend so much of our time really focusing on this one ecosystem within the vast part of the ocean? An ecosystem that only takes up 2% of the actual space within our ocean. And that's because coral reefs are so important, not just biologically, but to humans. So biologically, they're one of the most diverse ecosystems in the world. They rival terrestrial rainforests, and that's why they're called rainforests of the sea. And it's estimated that 25% of all marine life rely on reef ecosystems during one stage or another of their life cycle. But these reefs also provide a lot of human services that we call ecosystem services that we kind of take advantage of on a day-to-day basis. One of those is that they absorb wave energy and help to protect our shorelines. So as waves roll through, once that wave hits that healthy reef, hopefully, area, that wave starts to break apart and a lot of the energy is dissipated before it hits land. That may not be as important here in places like Saba. We have a ton of elevation. When you think about places like Florida where it's completely flat, that reef helps to protect a lot of our infrastructure, our roads, our hospitals, our schools, our homes that are all built along those shorelines. So they're incredibly important. They also provide a source of novel compounds that we use to fight human diseases. So right now we harvest organisms that only grow in coral reef ecosystems that create compounds that we can use to fight cancer, drug-resistant bacteria, and even Alzheimer's. And finally, at least 1 billion people around the world rely directly on coral reefs. So that's almost 1/7 of the world's population. And they rely on reefs for food, so protein from the fish, the coastal protection we talked about, cultural practices, and income, primarily from the ecotourism or fishing industry. So they're incredibly important to our economic foundation, not just locally, but globally. Now, before we move on, I want to talk about what corals are, because they're not as simple as sometimes they look kind of from the outside. So if you kind of dive underwater and you see a vast coral reef covered in corals, what you're actually seeing are living organisms. They're made of animals that kind of encrust a calcium carbonate skeleton right here. So very similar to us, we create a skeleton, similar composition to what corals create on their foundational skeleton as well. And that layer over the top is the animal, and it's a series of interconnected polyps with tentacles that can kind of wave out into the water to help trap food. But it's not just an animal. It's actually an animal in a complex consortium of microbes that interact with the coral animal to help make it healthy. One of those microbes is called the Symbiodiniaceae, which is also called zooxanthellae. These are tiny single-cell algae that live inside the coral. They're critical for that coral to survive because they provide a lot of energy and resources for the coral animal to live, and then the coral provides a nice home for it. So they have this great mutualistic relationship, and they actually can't survive without one or another. Now, when corals are stressed, and when they are stressed, they actually expel those algae out into the environment and lose the beautiful coloration that you normally affiliate with corals. And that's a term called coral bleaching. And that's an important phenomena. But today we're talking about infectious coral diseases, which is a little bit different. And that's where the second microbial consortium comes into play. There are a series of bacteria that work within the coral tissue, on top of the coral, and actually in the coral skeleton to provide a lot of resources to the coral. They can be probiotics for that coral animal, just like we have healthy bacterial probiotics in our gut. There are healthy bacteria that live within the coral as well. But there can also be pathogens out there, those nasty bacteria that can cause infections into a coral. Like us, corals are animals. They have an immune system, and they can get sick from bacterial infections, viral infections, fungal infections, just like we can. And that's the infectious disease component that we're going to be talking about today. Now, when you think about coral reefs, this is kind of the iconic trajectory of coral cover that's embedded within, our understanding of today's coral. So this is actually representative of the entire Caribbean over the last 50 years, where 50 years ago, in the late 70s, early 80s, about half of the coral reef was covered by living coral cover, and that's what the reef should be looking like. However, there is these series of events that have caused dramatic decline in that coral cover. And the first one, the most dramatic one that caused over 50% loss, was associated with an infectious disease called white band disease. This hit primarily the branching corals, the staghorn and elkhorn corals, which dominated the shallow reef environment throughout the entire Caribbean for at least the last 200,000 years. However, within the span of a few years, we lost over half of our coral cover because of this infectious disease. We think it was a bacterial infection, but we're not quite sure. There weren't samples preserved. We didn't have the technology we have today to really understand what that disease was all about. But since then, there's been a suite of other losses associated with really warm water temperatures, which we see in a cyclical pattern with El Nino events. Those are the ones that cause the coral to bleach. But remember, this is a disease story. And as all of that decline was occurring, a plethora of other different types of diseases have been identified. Now you can see these have very kind of unimaginative names associated with them. One of the reasons is because we are very poor at identifying marine disease pathogens. It's really difficult to grow bacteria on land in a petri dish that normally live on a coral. And so because of that, it's very difficult for us to fulfill all of the kind of steps that we need to identify a particular pathogen. So we have these generic names that describe what we see underwater, like white plague or white pox. Now, the take-home kind of visualization I want you to get from this slide is the healthy coral tissue are the ones that are colored, right? They're like the greens, the oranges and browns, the kind of tannish colors. The white area and here, this black band, which we'll talk about later, are the signs that the coral is sick. And they're not bleached. In a bleached coral, the tissue is actually still alive and it can recover. It's just white, it's translucent. When the coral is dead from, or is showing white from a disease agent, that tissue has sloughed off completely into the water column, and you're seeing the skeleton completely exposed. So it'd be like if my arm was a coral and I got an infection on my fingertip. As the disease moved through, my flesh would fall off, and you just have my skeleton exposed bright white. That's basically what's happening in these images here. So that one is a little bit different. Kind of complicated, but so the healthy tissue is here. These dark spots they've been kind of referred to as a bruising, but it's actually an upregulation of melanin within the coral. Melanin is part of the immune system of a coral, and it's actually part of the immune system in humans too. So when you get exposed to sunlight and you get that UV damage, you get an upregulation of melanin in your skin, which makes your skin darker. You get tan from melanin. Melanin is also found in corals, and that's what we're seeing there. There's a bacterial infection, and so it's upregulating this melanin as a protective mechanism to try to get that bacteria to kind of go away. But that's a great question, because that one is stand out. So all of these kind of diseases have been working together to really change the structure and function of coral reefs, not just around the Caribbean, but around the world. You know, so if you visited, this is Carey's Fort Reef, but honestly, this can be a picture of any shallow water Caribbean coral reef. In the 1980s, it would look like this, right? Staghorn coral in the front, elkhorn coral in the back. And if you visit these reefs now, you know, you get a very different image. These are skeletons of corals past. You see some of that structure there, but it's mainly covered in sediments and algae. Sometimes you see some of those soft corals come through, but those soft corals are not providing those ecosystem services that we talked about at the beginning, right? And so it's a very different kind of phase in which most of the coral reefs today are facing, and they're functionally dying. They're not providing the same services as what they used to 50 years ago. So we are working with the last vestiges today of what has survived all of these issues in the past. And so what we have out there is really critical to preserve and save so that we have individuals to recover from in the future. And to kind of make matters worse, we've had obviously diseases in the past that we've talked about. And before 2014, those diseases were all I studied. But now, since 2014, my lab has primarily focused on stony coral tissue loss disease. So it began in 2014 off the coast of Miami, right around here, and has since spread throughout the entire Florida Keys region, moving up to the entire northern part of the reef tract and down south throughout the Keys. And now, today, it's throughout the entire ecosystem, even into the Dry Tortugas National Park. So it looks like this image on the right here, where you've got the healthy coral, and then this white, newly exposed skeleton from the infectious agent moving across. And then this is like exposed skeleton that has some algae that has settled on it, but has died probably within the last couple weeks. This disease affects over 20 different coral species, mainly the boulder and the brain corals. The pathogen may be a bacteria or a virus, and there's evidence for both, and they could be working together. We think bacteria play an important role because, as I'll get into, this disease responds to antibiotics. So you can actually treat it with antibiotics and stop the disease from progressing, suggesting that bacteria play an important part in the tissue loss that we're seeing. Maybe not the primary pathogen, but definitely contributing to that tissue loss. We do know that it's a waterborne transmission. We've been able to model this throughout the environment, showing that hydrodynamic currents actually can predict where the disease moves within a reef scale. And within a laboratory setting, we can cause disease just through water moving from one coral to another. So water is the primary mechanism that this disease is transmitting within and among reef environments. Now, it looks kind of like this through time. So this is a great time series. This is a brain coral that was infected in late August. So you can see the lesions are kind of this conical shape that's moving out through time. And this entire coral, which is probably a couple hundred years old based on its size, dies within just a couple months. You can see how incredibly virulent this disease is and how quickly it can move throughout the entire coral colony. And unfortunately, it has not stayed isolated to Florida. It has spread throughout much of the Caribbean. So this is a great database. It's an open access database where people from around the Caribbean can enter data that's reviewed by a consortium of experts. All the red dots show confirmed cases. The purple are ones that are being reviewed, and the greens are ones showing no disease signs. So you can see it's, spreading throughout much of the islands, throughout the Yucatan, and kind of continuing through to move south over time, which is against the prevailing currents, probably associated with some human activities, perhaps ballast water. Now, if we take it a little bit closer into our local region, you can see, obviously, this area is, all identified as having positive stony coral tissue loss disease associations. SEBA actually has one entry. I put it up here with its complementary photos over here because this is the one data point within this global access information, which was really important for us to know before we got here. We've been able to dive core gut and tent shallow and saw that it was present there as well. You had a question.
Oh, I got it answered. I wanted to know what the ballast water was.
Okay. So for those of you that don't know, ballast water is what is kept in the bottom of the boat in order to keep it, weighed down during transit. And so there are regulations associated with how and when this happens, and sometimes they follow them very well. Sometimes it doesn't appear that they do. That is one hypothesis that is out there. So there's a lot of study behind it that's happening right now that suggests that may be the case, but it could be other mechanisms as well. Now, one of the novel things, as I mentioned, that has come out of this major crisis is kind of a phase shift in how we deal with corals underwater that are sick, because for probably about 15 years I studied coral diseases and I had no medicines to treat them. So I tried to figure some things out on my own. This is some work that we did in the U.S. Virgin Islands, actually at Buck Island Reef National Park in St. Croix. And this is a disease called yellow band. It's a nice disease to work with because it moves really slow. So I could try a bunch of things and see what worked. So we tried shading the coral because we knew light was important. We tried aspirating the coral, which basically sucked all of the diseased tissue up, but that was a huge pain in the butt. And we also tried chiseling, which was like a fire break, creating a fire break between the diseased and healthy tissue. And the fire break seemed to at least buy us some time, but none of this really worked well. And none of it could be done at a very large scale because it took forever to do. Each coral probably took like, I don't know, half an hour to at least get this application. So it wasn't scalable. So where this led us to was, reaching out to novel scientists that maybe had answers that field ecologists, didn't have in our back pocket. So I know a lot about corals, and I can identify coral disease, but I don't know anything about medicine. So during this crisis of kind of the realization of stony coral tissue loss disease, our community, our scientific community, reached out to this group that had formed called Ocean Alchemists. It's a two-person group. They're pharmaceutical scientists during the day, and they heard about this crisis with stony coral tissue loss disease and thought, well, maybe this can be our night job. And that's basically what happened. They started working with us scientists saying, okay, like, what can you tell us about the disease? What can we do to help? And during this formation of a collaboration between a group of scientists and these pharmaceutical scientists, we discovered a couple things. One, you could take the corals out of the reef and put them into an aquaria and dose that aquaria with antibiotics, and it stopped the disease from progressing. But you can't dose the ocean with a bunch of antibiotics, right? Who would want to do that? That would be very unhealthy also because there's bad things that come out of antibiotic use when you're not targeting it directly, which we'll talk about. So what they did was create this underwater ointment called Base 2B. And I don't know if you've ever thought about it, but imagine trying to take a lotion underwater and spread it onto something that's slimy. It's not easy, right? So they used their knowledge base from their pharmaceutical scientists to create an ointment that basically can stay together underwater. When you spread it on the coral, it adheres to the mucosal layer of the coral tissue and also the skeleton. It creates direct contact with that mucus and allows anything that's within the ointment to transfer directly into the coral tissue. And it's customizable, so you can integrate primarily amoxicillin into it and change the release rate over time. So say you want that release rate to be quick and strong, you can adjust the formulation so that it gives a high dose of antibiotics within a day, or you can slow it out so you can have a 3-day release period over time. And this ointment is customized for this particular antibiotic, so you can't use it on, a bunch of other things that maybe you think will work. It's very specialized. And this is what it kind of looks like underwater. So this is a treated coral. Now we're moving on to a coral that has stony coral tissue loss disease. You put the ointment that's mixed with amoxicillin into this plunger syringe. You take it underwater, you squeeze it out, and as you squeeze it out, you press it along the disease lesion. And so it's super quick to do underwater and highly effective. Actually, studies have shown so far that this Base 2B and amoxicillin combination here is 84% effective in halting the progression of stony coral tissue loss disease. So they tried, not treating the corals, everything died. They tried it just as a couple different new bases that were, different than this Base 2B, primarily shea butter, which can be used sometimes underwater. Those didn't work. Just adding Base 2B didn't work. But the Base 2B and amoxicillin was a success. And so this has been widely accepted and used in a few different places, primarily Florida and the U.S. Virgin Islands, to treat stony coral tissue loss disease. And this is just a glamour shot of Dr. Williams here, underwater, using, sometimes it's hard with the plunger, so we use a caulking gun to make our lives easier and reduce the pain and strain on our hands as we are treating the corals. And we put these nails in so that when we revisit the coral, we know where we treated, and we can see if the disease has progressed from those nails or if it has halted at the treatment area. And then we, of course, take those nails out when we're done. So in Florida alone, over the last probably 4 years or so, we've treated over 22,000 corals, and most of it is done by a consortium of what are called strike teams throughout the region. And these strike teams get enabled when disease is visualized on the reef. They can go out, do their treatments, follow up, make sure they're effective, and they can do it throughout the entire Florida region. And this example of a treatment strike team intervention strategy has also been translated into places like the U.S. Virgin Islands, where all of these stars represent where these treatments have occurred in St. Thomas, St. John, and St. Croix. And they've successfully treated over 17,000 corals within the region as well.
Can we do an applause for that?
Yes! So obviously, Dr. Williams and I are a part of this group. We're studying it. We're trying to figure out more, what are the benefits of changing things a little bit? What other alternatives can we use to kind of add another tool in our tool belt? But this, these are, as you can see, there's hundreds of dives and hundreds of people working on this collaboratively together, and we're just kind of representing a bunch of the efforts of the consortium. But yes?
Yeah, so the antibiotic and the ointment that we're using, it stops the spread of the disease.
Mm-hmm.
Over time, would it recover? Does the coral kind of heal itself and cover the area where it was infected?
Yeah, so if you can stop the disease from progressing, that coral tissue over time can regrow. And it won't necessarily always regrow over the area that died, but it will grow like up and out, So it doesn't always, their morphology is interesting because they don't always have to, recover what they've lost, but they will continue to grow and they will create more tissue through time. Mm-hmm.
How long-term is the effect of the antibiotic?
Yeah, we're still trying to figure that out. But the great thing is that, once the wave kind of moves through, you're at a slow burn, right? So when the wave first moved through Florida, it was beyond control. But at this point, we've got corals that are left, and you can kind of hit the flare-ups over time, and it's manageable on a local scale. So, yes.
So, when you treat them,
Mm-hmm.
does that in some way stop it from spreading?
It absolutely will, because as the coral is dying, it's releasing diseased tissue into the water column. And when that diseased tissue flocculates off of the coral, it transmits to its nearest neighbor. And some of our work has actually been published to show that corals within 2 meters of a diseased individual is more likely to get sick through time. And so as you can treat individuals that are sick, you're reducing the pathogenic load within the environment and potentially having effects on other corals that are not treated. But there are, of course, challenges with using antibiotics to treat stony coral tissue loss disease or anything. One is that the cost of antibiotics is a lot. So it could, if you're trying to kind of have a couple days of work, you could blow through hundreds of dollars of antibiotics. And so it's costly. Sometimes the accessibility of getting antibiotics is actually a big challenge. Sometimes we can get antibiotics easily. Sometimes we have to go through a certified vet. Sometimes it takes days to weeks to get it in the mail. So it's difficult to get. And the company that you buy the base 2B from, Ocean Alchemists, can't give you the antibiotics. You have to actually get them on your own. So it just makes accessibility more difficult. And then you are, inherently, we've known through a lot of human uses of antibiotics that high use of antibiotics is creating antibiotic-resistant bacteria. So you don't want to use too much because then you're over-selecting for maybe something that is more virulent. So there's a lot of these, decisions that you have to make when using, right now, the only, treatment method to treat and effectively stop disease from progressing with this particularly virulent disease. However, we are working with Ocean Alchemists to create a non-antibiotic based option. And these are what they're calling their coral cure ointments. These are new natural-based product formulations. They're non-antibiotic. They have antiviral and antibacterial components with them, but they're all naturally sourced materials. So they can actually customize it to have different types of active ingredients within it. They can adjust the release time, and they can actually create everything for you and mail it to you, so it's plug and play. Right? So you reduce like the shelf life of having to worry about antibiotics, you reduce the costs, and potentially have a better way to treat, corals with something that's, a non-antibiotic based.
Excuse me. The amoxicillin had an 84% with base 2B, had an 84% cure rate, I guess?
Yeah.
How do these compare?
So that's what I'm going to talk about next. Thank you. Great question. So we're actually right now testing 4 different types of coral cure products. And so we're testing them right now in the USVI, and then partners of ours are testing them in the Turks and Caicos and also the Cayman Islands. So this is kind of an example of we come across a lobe star coral, and it's got a bunch of different lobes that are infected, and we can try these different types of formulations on the different lobes. And then Sarah's actually going to be going back next week. She leaves directly from here to go back to the USVI to assess the efficacy of these 4 different applications on the treatments that we did a couple months ago in Virgin Islands National Park. But some of our preliminary data show some promise. So this is just an example of a study we did a few months ago in the U.S. Virgin Islands. We've got corals that are controls. That means we don't give them any type of treatment. We have corals that have what were called Coral Cure 2D, and then we've got Coral Cure 3D here. And I call it these like random names because as part of the relationship, they are developing proprietary information that they are going to commercialize, and we are their blind testers. So they send us products, and we test them blind so that we are not biased in our interpretation of what we're actually like collecting within the data. So we kind of have these random names associated with the treatments. When we look at progression rates, so this is how much coral tissue was lost. We've got different species at different sites here, and the colors match up the colors around the picture. So the purple are the control, the orange is 3D, and the green is 2D. And I just want you to focus on these areas here in the boxes where the 3D in the orange shows much less tissue loss compared to no treatment. And this is showing us that there's some promise here. So they reformulated this a little bit, and that's one of the things we're testing right now to optimize this effect. But it's still about 60% efficacy compared with the 84 of the base 2B. So we're getting there, and we think we'll get there. It's just we need a little bit more time. Next week. Yeah. So that's some of our next steps is like, can we find that natural-based product treatment that's as effective as an antibiotic? And that'll give us a lot of different like tools in our tool belt. And then we're actually measuring the effects of the treatments on coral microbiome. So as we're treating corals, what bacteria are actually changing? So we're getting like before samples, after treatment samples, and we're comparing that through time. And we're actually measuring the concentration of antibiotics in the base 2B to see if we can reduce that, which will save cost. It will reduce the probability of antibiotic resistance. So we've got a bunch of different trials right now where we're reducing things by halves and quarters and looking at the efficacy through time. Now, I just want to touch upon one more disease. And this disease is kind of been, hasn't been up in the news very much, but this disease has been around for decades, and it's a global disease, and it's called black band. So this black band right here is a cyanobacterial mat. It's a consortium of bacteria that work together to create this toxic environment to the coral tissue. So it kills the coral tissue and then eats it as it moves across the coral colony. And because this is pervasive, it's global, it's another really important disease that I've been studying for a while, and I thought, well, let's try to build off of this novel partnership and get a twofer, and then we'll keep going more. So this study was, again, our objective was to create a method that could treat this black band disease within the water, like at the reef site, that could be shared and implemented around the globe. And this study took place in the U.S. Virgin Islands, in the northeastern part of the Caribbean, off of St. Croix. Buck Island is the northeastern part of that island. And then off of Buck Island, we're on the northeastern part of the bay. And so right here, all of these dots represent corals that had black band disease. So there's like very prevalent within this region. It's killing corals that are probably 400 years old, if not more. And it takes about a few months to a few years to kind of kill these corals off. So I thought, well, let's work our magic and figure out what we can do with this disease too. So we had 3 separate series of treatments, and we would treat the corals, what we call at time 0, and then we'd go back 3 to 6 months later and look at the efficacy. We would measure whether that black band disease was still there, and we would measure how quickly it moved through time. So here's that nail again. We'd take these measurements to the black band and look at how much tissue had been lost. So our first series of treatments integrated a natural product called hydrogen peroxide. It's a common antiseptic. We use it to treat, wounds, to kill off, potentially infectious bacteria. I thought, let's try this underwater. So we had this high, food-grade hydrogen peroxide that we would dose onto the skeleton. We also figured out that hydrogen peroxide is in tooth whitening gel. So if you want to whiten your teeth, you're actually putting hydrogen peroxide on your teeth to make it whiter. So I was like, let's try that. So we did. We tried it with epoxy, and we tried it mixing it with the base 2B. And we also tried the base 2B with amoxicillin because we thought, why not? This works with stony coral tissue loss disease. Maybe it'll work with black band. Our second series of treatment were some of the coral cure ointments that Ocean Alchemists created, and they integrated this Lugol's iodine solution within 2 out of those 3. And that's a prophylactic that we use often to prevent diseases within corals that we're culturing in aquarius settings. So actually, like people that have home aquaria are very familiar with Lugol's iodine solution as a way to keep corals healthy. And then we had urea oxide, which is a more stable form of hydrogen peroxide, integrated as active ingredients in these coral care ointments. However, one of our biggest issues was adhesion. Like, I can't tell you how difficult it is to stick sticky things to corals and make it stay there. So this would take me, like 10 minutes to get on, and then I just sit there, watch it fly off. So adhesion was the bane of our existence. So I worked with Ocean Alchemists to come up with an alternative. And this is what we called the rope trick. So we took hemp-based rope, it's called jute rope, which is a kind of similar to hemp, and we saturated it in the coral cure ointment. And then we took this really crazy tech and we stapled it to the coral using an underwater stainless steel staple gun. And so we'd lay the rope on the black band and then staple it across the coral colony. Now, one of the things we found was that if we just did 1 layer of the band, as we removed the rope, the black band would move so fast, it would eat the coral so quickly that it wasn't effective enough. It wasn't working. It wasn't thick enough. And so we integrated what we called the double wide. So basically taking the rope and making it 2 layers so that we could have more, coverage within the application. And then we integrated a few more of these now proprietary natural-based product blends of the ointments. We also integrate some controls. So when we're always trying these things with active ingredients, we want to make sure that the delivery mechanism isn't the reason for success. So we have just plain epoxy, just plain base 2B, no treatments, and just the rope without any ointments. And I'm just going to walk through the results. So we had the series 1, totally ineffective. Nothing worked, nothing at all. So that was pretty disappointing. Series 2, totally ineffective. Nothing worked. The black band just like ripped through our treatments and all of the ointments that we applied. However, we got gold when we tried series 3. So this is again the progression rates. Here are the controls with no treatment, and then here are our double wide coral cure ropes. 3 different types of cocktails, a D, E, and an F. All of them were highly effective, but these 2 were the winners. And if you kind of look at it through time, you've got a coral with the black band here. You've got the double wide rope covering that band here. This is 1 week. We removed the rope from the coral, and then the coral is healed, and it stayed healed for months after. And this coral now can regrow over its tissue lost or continue to grow as a colony. And so some of the conclusions here is that these coral cure ointments not only do they have hope for stony coral tissue loss disease, we can use them potentially to treat a global, worldwide coral disease black band. They are natural-based products that contain antimicrobial and antiviral properties that could be much easier to send and distribute around the world compared with antibiotics, which are highly regulated. Probably decreases that risk of creating antibiotic resistance within some of those large-scale treatments that we've got going on with stony coral tissue loss disease. And this may be more reef scale approachable compared with SCTLD because black band is kind of seasonal. We see it a lot in the summers. It's more slow. It doesn't rip through the colony like stony coral tissue loss disease. And it actually has depth restrictions, so it's in shallow reef environments. So this may be a really strong management application for the community to use to make targeted decisions to treat coral disease outbreaks. And then finally, I just want to kind of wrap it all together that, you know, obviously the coral reef is facing a major crisis, and we are racing against time because we are working with the last colonies left that have survived the 50 years of stresses that have gotten us into this situation. But those kind of stressor events and those crises have led to really significant novel partnerships that have created greater arsenal to combat global diseases. And these disease outbreaks has really caused a ton of momentum in the scientific community and in the local community to get people motivated and interested in preserving coral reef ecosystems. But every community must decide what's appropriate for them. And there are risks associated with bringing medicines into the ocean. And so that has to be, you know, calculated risk. And you have to weigh the benefits and the cons associated with that. But now we have tools. And now we have choices where before we didn't. And I'm super excited that I got to be a part of that. So that I would like to thank you again. Thank you to our sponsors. Thank you so much to the See and Learn Foundation. It's been a pleasure, and I'm happy to answer any questions.
Do you see this as something that, say, the local marine park personnel could be trained to do in their own park?
Absolutely. It is not difficult
Repeat the question.
Oh, repeat the question, yes, for the online audience, which hello. Is this a tool that the local marine park staff members could consider integrating? And yes, it is. So it's definitely an option for the local management agencies to consider and using. It's definitely been implemented in different places around the Caribbean. So it is a potential conversation to have to see if that's a choice that the community wants, right? Because it has to be, you know, a local decision.
Question related
Yes.
to that.
Yes.
Who's funding your work and who's funding, what is it, Ocean Alchemists?
Yeah, Ocean Alchemists, I don't even know. They're just doing it. I try to give them money sometimes, and they are just really, I don't know. I think they've got a couple philanthropic partners, but they're really just doing it. They're figuring out how to do it in their house and in their basement and with stuff. Yeah. Yes. So who funds us? For this project, the National Park Service funded us. So we wrote a project with them to specifically develop treatments to treat coral diseases within the national parks. And we just applied for an EPA grant to help bring some of our work forward a little bit more. So we're hoping to hear within the next month whether we can keep moving this forward. Yes.
So you see that with these treatments, the corals are saved from disease long term, but does it prevent the corals from being reinfected in the future?
Yeah, that's a great question. So it doesn't. And repeat the question. Thank you. So once the coral is treated, is it completely cured in the future? Can it get reinfected? And it can get reinfected. And what we sometimes see is that you treat one lesion and another one comes up. But that doesn't always happen. And I would say the data from Florida suggests that that doesn't happen most of the time. So I think we're still trying to figure out what happens within the coral when it's treated. But corals can share resources because they're an interconnected colony of polyps. So they have channels that move things from one side of the colony to the other. So what you do over here can certainly influence what happens over here. And we're still trying to understand how that works, how much of an influence we have, and how protective it can be long term. But that doesn't keep it free of disease forever, right? So it's a great point. Did you have a follow-up question?
Yeah, like it's very small. I don't want too much time, but then I mean, depending also on how the immune system of coral reefs work, like is there possibility of some sort of vaccine or some sort of way of teaching the immune system to respond with some B cells or T cells, something like that, to the disease?
So the question is whether you could kind of treat the coral with a vaccine to kind of give it a memory of disease, right, and have it stronger, or have the ability to get exposed and then fight the disease in the future. And corals don't have an adaptive immune system, so that means they can't learn from exposure. However, there's some evidence to suggest that there could be some immune priming that happens after a coral is exposed to disease. And that's kind of at the edge of like cutting edge place of where we're going, you know, is like, can we immune prime it somehow using the innate simplistic immune system that corals do have? Yes.
Have you noticed any negative effect of any medication to the wildlife around the coral?
So the question is, have we noticed any negative effect to the wildlife of the reef after treatments to the coral? And that has not been documented, or it's not been observed. So I would say we see fish eating, picking at it when we initially put it down, but it doesn't seem to have any adverse effects to the fish. However, I think there needs to be some long-term studies to really understand that. But so far, there's no evidence of that yet. Yes.
So you had so much success with pharmaceutical scientists. Are there microbiologists that are trying to identify the pathogens? And
yes.
Are you working with them in any way?
Are there microbiologists that are working to identify the pathogens? And yes, there are a suite of them. And we actually have some studies out where we will look at the molecular approaches for the bacterial community, but we also collaborate with culture-based microbiologists that can grow a subset of the bacteria in petri dishes. And actually, while we were here, some of the corals that we tested that showed resistance to stony coral tissue loss disease, our staff back at home was just draining some of the mucus off of them to get it cultured to figure out if we could identify the probiotic that could be potentially making them more resistant. And they have libraries upon libraries of bacteria that they're trying to use to maybe identify the pathogen, but so far it's been years and they haven't kind of unlocked that one yet.
Erinn, I might have a good follow-up to that, which is you mentioned 2D and 3D had antivirals as well, so could this be sort of a commensal bacterium that itself has been infected with a virus that is now making it pathogenic to the coral, which will complicate the culturing, right?
Right, so the question is there a viral component to the infection or to the pathogenic infection of stony coral tissue loss disease that could be complicating the process? And yes, and there is some evidence actually that the algal symbionts that live inside of the coral tissue potentially could be getting infected with a virus, and that virus is compromising those algae. The coral then is more potentially susceptible to this commensal bacteria, or the saprophytic bacteria that may be opportunistically coming in and being the agent that causes the tissue loss, where the initial infection is caused by a virus. So we have probably 250 of us at this point in time, all working together to study different components of the disease, etiology, how it spreads within the environment, and treatments. So that has been another positive outcome, is this open access kind of regional, if not national, network of scientists working with managers and coral restoration practitioners and everybody to try to understand this disease as best as possible, and then also provide kind of a, this is what we did example in Florida that we can, you know, give to our partners around the Caribbean as this thing spreads, is like, here's an example of what we did. Figure out how, you know, what works for you and what doesn't. Yes.
This is great, really exciting, thank you. I noticed that Ocean Alchemist was an LLC, and I'm wondering if they're talking about kind of monetizing that when they track it eventually, and if so, are there any discussions about how it would be made available to communities without many resources?
Yeah, so the question is Ocean Alchemist going to be monetizing their products, and is there any potential offset associated with making them kind of more available? And yes, like, so they are definitely like, you could buy these products online right now. I mean, they have to like make sure you're credible, because they won't give them to anybody. You know, they won't give them to just random community members. So you kind of have to go through this vetting process. But these ointments are going to become commercially available, but they are extremely generous with their time and with their product. And if there's communities that don't have the funding, they still do it. I mean, they just hand out products because they're like, please try these. Like, tell me what works, what doesn't work. And like, they want to do everything they can to help. Yes, they are a business, and that's why their information is proprietary and they're protecting themselves in that way. But I also think as scientists, we have to figure out how to work well with groups like this that, you know, have to protect their commercial IP, but they're the ones that are going to be moving like our conservation efforts forward at an accelerated pace.
Are the lotion preloaded? So I saw that you have syringes. Are they preloaded from the factory, or we can do them? We can load them as local site.
So the question is, are the ointments preloaded, or do you have to do it themselves? And it depends. So if you get the base 2B and the amoxicillin, you have to mix it yourself. So you have to weigh out the amoxicillin, weigh out the base 2B, get the right formulation, mix it yourself, and then load it into the syringe yourself. And then you only have 24 hours to use it. So there's like shelf life. Now the other ones, the Coral Cures, the cool thing about that is they do it all, and they'll send you the prepackaged, preloaded syringes, so they're like plug and play. And they have a much longer shelf life, I think probably months, I would guess. Plastic waste? Yeah, you can reuse them. So you could send it, I'm sure you could send it back and reuse them, but yeah.
First and foremost, thank you so much for this presentation tonight. To the weekend warrior marine biologists or citizen scientists, are there any pathways of communication to report the progression of this disease to authorities, to scientists, so that we can better help to understand where it's going?
Yes. So the question is, as a citizen scientist, when you're out in the water, is there any way to report the disease to a greater database, kind of? And there is. So the map I showed you, and I don't have the website, but it's AGRA, A-G-G-R-A. It's a network of scientists around the Caribbean, and it's completely open access database, so you can actually upload your information there with a couple pictures, and then they review it, and they'll probably reach out and say, so, you know, did you see any others like this, or, you know, ask you some clarifying questions. But that is where we're actually entering all of our data that we collect while we're here into this regional repository, so that it's open access for everybody as well. So before I close, I'll just say, if you are interested in trying to treat some coral diseases, we have some diseased corals with some ointment over here. And it has been a lot of fun doing it with the community members this week, so we thought we'd bring it over in case you guys wanted to get your hands dirty. We also have a 360 underwater video that shows what it looks like when we're treating the corals and taking samples, which will be cool. And then there's also some swag over there, like a bottle opener and a magnet in one. So if you want some swag, come over to the table, and we'll be happy to help and keep talking to you all.
Maybe tell a little bit about what you're going to do on your field project tomorrow.
So tomorrow in our field project, if you want to come diving with us, which you should, because it's going to be awesome, we are going to be doing some underwater roving coral disease survey. So one of the information that gives us the most, you know, data that we can use is how many corals are out there right now in your 30-minute swim, and are they healthy or are they sick? And what are they sick with, and what does it look like? So it's all the data that we can upload to the AGRA database, and we'll be able to teach you guys, you know, what these healthy corals look like, and be able to identify what sick corals look like, Larry was out there with us the other day, and he became an expert in about 30 minutes. So you can become an expert too. So please come join us tomorrow. Thank you.
Another big round of applause for Dr. Erinn Muller. All right, we'd like to thank everybody for coming, and if you are staying for dinner, it will be ready in a little bit. We're going to clear out the chairs, so if you want to help us, that would be great. Bring some tables in, and everyone have a good night, and we will see you tomorrow or next days. Yes.
Miami is known as an international community and the hub of trade and logistics for the Caribbean and Americas. A leader in the freight logistics industry, CaribTrans has served the Caribbean region for more than 30 years, with a footprint that has grown from one island to now serving the majority, as well as Central and South America. CaribTrans is an NVOCC, a non-vessel operating common carrier, meaning it has the same responsibilities as a shipper without owning the vessels or planes. Its primary customers, individuals who ship clothing, electronics and other personal items, and occasionally cars.
Most of the stuff that they're sending is because they don't get it there. So for us to provide it on two services, air and ocean, we can give it a choice of how fast you want it. So if you want it really fast, we're going to send it by air, you're going to have it next day, sometimes even the same day.
That on-time service has been the key to CaribTrans' success, whether shipping by air or by sea. The company moves about 5,000 TEUs of freight each year, more than any NVOCC in the region. And since it joined the Saltchuk family of transportation companies a couple years ago, CaribTrans can offer customers even more shipping options.
Other NVOCCs, they depend on other shipping lines that are not part of their family. So they need to rely on whether they're going to sell or not, whether they are late or not. It doesn't happen to us because it's part of our family of companies. We control the service that we give.
Sí, buenos días, por favor, con Diana.
Quality service is what keeps customers coming back. Customers are treated like family, and when they leave to research competitors, it doesn't usually last long.
They have left and they have come back because they've said that, nobody does it better than CaribTrans. So because of that relationship.
That exceptional service includes consolidation. Customers can have freight sent here and stored for free for up to a month, then consolidated and shipped on to the islands and Americas, a perk for the region's growing small business market. Puerto Rico is an expanding market for CaribTrans, about the size of Connecticut and rich in history and culture. Puerto Rico is home to many pharmaceutical manufacturers, as well as a thriving tourism industry for which CaribTrans moves raw materials and supplies. But serving small businesses is still the company's bread and butter here in San Juan. Businesses like Caldera Café, a local coffee company. Owner Alfredo Rodriguez started growing coffee beans at his mountain farm about 20 years ago. He recently ordered a variety of new equipment for his farm and shop. He uses CaribTrans because it consolidates shipments, which cuts down on the number of times he has to pay the Puerto Rico import tax.
Not many companies want to do that. They want only big cargoes and maybe half container or full containers, but they don't want to consolidate because it's more difficult.
Turning challenges into opportunities is what CaribTrans does, and that includes helping to grow small businesses like Caldera Café by taking the hassle out of shipping.
We can be a gateway to expand their business in the Caribbean and back to the U.S. And wherever they can sell their products, we are also going to be a partner for them.
That customer dedication has made CaribTrans what it is today. Now as part of a larger family with more transportation resources, CaribTrans hopes to grow beyond its current market, offering services to additional Latin American ports and eventually Europe and Asia. But even when it becomes a global logistics company, CaribTrans wants to stay true to its roots, always providing personalized, reliable customer service and a commitment to the communities it serves.